Yeah, so I'm very pleased to come here for this presentation. Today, I'm gonna actually tell you two stories. The first story is on Dynamo for predictive modeling for the single-cell genomics. And then I'm gonna also talk to you how we can use Stereo-seq to perform advanced spatial transcriptomics. As you can see, both work have been recently published in Cell. So yeah, so for many years, I have always interested in moving towards predictive modeling of single cell or for biology in general. So I want to start with a very theoretical view to motivate how I'm thinking about biology. So in the discovery of the fundamental principles of astronomy, 400 years ago, Tycho made the first comprehensive observation of the planet movement, which leads to the derivation of the three empirical laws by Kepler, and then eventually the unifying theory of gravitation by Newton. So the key question is now with the big data in biology, can we also overturn unifying and also predictive theories in biology? Specifically, similar to astronomers who like to understand how the planet moves, also always wondered how does a single fertilized cell give rise to an entire organism with trillions of cells in a precisely also spatial temporally controlled manner. So now, as we know, we can use single-cell RNA-seq to generate all kinds of the cell atlases, and this really provides the unprecedented view of the conditions of the progenitor cells to all kinds of mature cell types across an entire organism. And it's still clear that biology is actually at this phase of moving toward to predictive modeling with the big data. But routine single-cell analysis are rather descriptive instead of predictive. For example, most of the analysis will just stop at cataloging different cell types by doing cell clustering and also annotation of the cell types with different marker genes. Recently, the pseudotime and also RNA velocity methods also gain popularity. So, for example, we can using pseudotime, we can then order the cells based on the transcriptome similarity across the cells, and also reveal the genes that are dynamically changing during the dynamic process. On the other hand, with RNA velocity, leveraging the incidentally captured intron reads, we can derive a measure of the time derivative of the spliced RNA or RNA velocity, and then we can visualize this RNA velocity to the low-dimensional space to reveal the local dynamics of each individual cell. Of course, those methods are very powerful and was very popular. However, all of them are very descriptive, and they also has a lot of fundamental limitations. So, for example, with the pseudotime ordering, it only tells you the simple trend of the cell fate progression, and also most of the time you actually need some post-processing approach to reveal the beginning of the cell fate condition. On the other hand, for RNA velocity, because it relies on the incidentally captured the intron, so the RNA velocity estimation is really unstable, and also it only tells you the local dynamics of each individual cells. And because of some underlying assumption, like the splicing rate has to be one for all the genes in all the cells. So the RNA velocity is relative and scaled by this gene-specific splicing rate. And also, as we starting to even more appreciate, which is that because RNA velocity assumes piecewise transcription rate, so oftentimes you actually get wrong or even opposite RNA velocity flow. So A few years ago, when I was thinking hard about this, and I realized that in order to solve all these problems and move towards to predictive modeling, we would love to firstly develop novel technologies that improve the RNA velocity measurements. And secondly, we would love to move beyond the discrete RNA velocity vectors, the local velocity vectors, to a continuous function. And it is immediately clear to me that once I have such an equation, so I can use it to predict the cell fate to each individual cell. And then we can also use it to resolve the underlying gene regulatory network, and even making non-trivial predictions, like predicting the optimal gene transcriptomic factors required for reprogramming, as well as making in silico perturbation prediction. So in order to achieve this overarching goal, we then tried to search and also implement an innovative technology to improve the RNA velocity measurements. We also developed this predictive framework, Dynamo, to make a mechanistic and predictive insights of cell-fate conditions. So Dynamo has four major innovations. So it starts with taking the single-cell RNA-seq data, including either the conventional, the regular 10x data, or the 10 resolved metabolic labeling enabled single-cell RNA-seq. And this later one will give us, as I will tell you later, will give us much more accurate RNA velocity measurements. And then it will take this data to estimate the kinetic parameters, like transformation rate, spatial, and so on. And then to learn the RNA velocity. But once we learn the RNA velocity, then we will move beyond this velocity vector to a continuous function to learn the vector field and couple it with the differential geometry to gain the underlying regulatory mechanisms and even making non-trivial predictions. So in the following, with the intention to demonstrate this four innovative steps in Dynamo, I'm going to firstly walk you through the ideal data and also experiments for learning such an equation. And then I'm going to tell you some theories related to differential geometry and using this to making predictions of, and also then some methods, and followed by some examples that I hope will be very illustrative of how powerful this predictive model can be. Finally, I'm going to close with the Stereo-seq for spatiotemporally resolved cell atlases. So let's start with the data. So it turns out that the ideal technology to improve the RNA velocity measurements seems to be the metabolic labeling enabled single-cell RNA-seq approach. And there are many different ways to do that, including one that I got involved with the SCNTC in with the work group at the UPenn. A very nice property or feature of this technology is that they can enable direct measurements of the next RNA. Thus, in principle, they will avoid all this biased measurements of the introns and also the unknown spatial rate from the conventional single-cell RNA data, because now we actually know how long we label the cells. So I believe this kind of data will enable us to not only improve the robustness of RNA velocity estimation, but also give us absolute instead of relative velocity, or velocity that actually has a physical meaning, like how much molecules are produced in a unit of time. So then, in order to analyze this kind of data, we developed a comprehensive modeling framework to estimate the kinetic parameters. It's the first step for RNA velocity estimation. So without going into too much technical details, here I'm going to just show you that we have, in general, 3 different models to model different data types. So for the first model, it was used to model the conventional single-cell RNA-seq data without the labeling. This is similar to the SC-value and the velocity approach. For model 2 and model 3, and there is no methods prior. So For model 2, we use we model the RNA labeling, but model 3 actually consider both RNA labeling and also the RNA splicing. So we also made great effort to base the model different types of the metabolic labeling experiments, including the one-shot kinetics and also degradation experiment, metabolic labeling experiments. So for kinetics, so you basically label the cell for different duration of the endpoint, and then you harvest it for the SC and TC. So for the sake of time, here I'm gonna only showcase how Dynamo can be used to accurately estimate the mRNA kinetic parameters for a kinetic experiment. So here I'm gonna use this UNG gene as an example, and I show you that we developed this two-step model to estimate the kinetic parameters. So you can see on this base plot of total RNA and the new RNA, there is a linear relationship for the new RNA to the total RNA, and then you can also see the slope of this linear relationship actually gradually increase as a labeling time increase. And if we then take this slope and they convert it into negative log 1 minus k and plot this as a function of the time, then you can see a secondary linear relationship, and that's why we call it a two-step model. So we take these two linear relationships to estimate the transcription rate as well as the degradation rate. And interestingly, we show that our degradation rate has interesting properties. Like if we plot the human's degradation rate and against the autolog genes from the mouse, and then you can see there is like a proportional increase of the degradation rate in the mice compared to the human, and this indicates the RNA turnover or the RNA degradation in mice is much quicker than in human, and this is consistent with the two recent Science papers where they show the protein degradation rate is actually much faster in mice. So now we're gonna also tell you how we can use the Dynamo leveraging the metabolic labeling data to overcome some intrinsic limitations of conventional RNA-seq velocity estimations. So here we applied the SC and TC approach to generate a data set of human hematopoiesis, and then we developed this new tool called the Dynamo to quantify the new total spliced and unspliced RNA for each gene in each cell. And once we have such data, then we can use the spliced and also unspliced RNA to quantify the RNA velocity. Here we actually use the state-of-the-art dynamic model from Velocitor to perform the RNA velocity analysis. And you can see the velocity flow is quite messy, and also there are some problematic conditions from the terminal cell level, like the megakaryocytes, urostory, basophil cells moving backward to the progenitor cells. On the other hand, if we use the new and total RNA to perform the RNA velocity analysis using Dynamo, then you can see automatically we obtain a very smooth hierarchical transition from the HSC firstly to the DMP and MAEP like cells, which then ramificates into like the neutrophil, monocyte, basophil, urostory, and megakaryocytes. And lastly, we captured the known biology of the human hematopoiesis. This indicates the labeling data with Dynamo's framework is able to correct a lot of problems from the splicing data. Another thing I want to highlight is because we do know we label the cell for like 4 hours, the velocity we get with the labeling data is absolute. So our method is very general and it can be applied to other data sets. So here I'm gonna also quickly show you that for this called mouse neuronal cell activation experiment, we can also learn beta RNA velocities. So in this study, the group from Japan dissociate the mouse cortex cell into the median, and then the fetus cell with the phase U, and then at a different time point, the trigger the neuronal activation with KCL followed by SCN2G. So when we use the labeling data to perform the RNA velocity analysis, you can quickly appreciate this nice transition from temporal 0 to 120 minutes with initial rapid induction from temporal 0 to 15 minutes with so you can see this the weight of the spin length or the speed of the velocity is actually very fast, and then it tunes down. However, if you use the splicing data, you won't see this kind of picture. So now I talked about some ideal data, the labeling data for estimating the RNA velocity. And then I'm gonna next tell you some theory behind our approach before I'm gonna explain our method for actually learning the vector field equation. So fundamentally, we use dynamic systems view to understand the cell state condition. So under this perspective, each cell can be represented as a point in a high-dimensional gene expression space, where each dimension, like X1, X2, X3, they are corresponding to different genes. And then the velocity of the gene expression, or the time derivative of the gene expression state X dot is governed by the underlying gene regulatory network function Fx. So this Fx essentially just represents the underlying gene regulatory network. In general, this is very complicated and involves thousands of genes. However, there may be some characteristic regulatory network motif, like the self-activation, nutrient inhibition network motif. And then if we know this vector field function F, then the trajectory of the cell in this static space or XT will be just following this vector field function under the deterministic assumption. And so in the next few slides, I'm going to use this toggle switch model I just mentioned to demonstrate how we can gain mechanistic insights with various different differential geometry and dynamic system analysis. So because we for this simulated system, we do know the underlying differential equation, so we can actually numerically calculate the vector field, and you can see in this vector field, there's three states, S1, S2, they're coming from different origin, but end up in the same terminal cell state or attractor state A1. And in fact, this system has three attractors. You have A1, A2, A3, and each of them is corresponds to a attractor basin like B1, B2, and B3. And this kind of definition, in my view, may provide like important theoretical foundations to understand what is the actual cell type, because for the flow within each attractor, they will eventually converge into the same attractor state. And the boundary lengths that separate the attractor basin are the separators, and on top of it, you can see different starter points, where the local RNA velocity flow has a height curvature. So next, I'm going to tell you highlight an important concept, RNA Jacobian, and demonstrate how we can use it to reveal cell state dependent gene regulations. So again, here we use this toggle switch model. And in plain language, Jacobian is just saying if how does the velocity of a gene changes when you change the expression of another gene. And the important thing is that you do need a vector field to do this kind of calculation, because even you can do the velocity accurately, you cannot take the derivative until you have a vector field equation. And here, let's focus on this specific domain where the X1 is increasing, but the X2 is fixed to be around 0.5. And then we can firstly plot the velocity of X1 or F1 is a function of X1, and this get this kind of curve. And then we can calculate the gradient respect to this name, which essentially gives you the Jacobian, or in this particular case, which is like a partial F1 or a partial X1, meaning if you change the expression of X, increase the expression of X, what will be the change of the velocity? And you can see immediately there is a strong positive peak when X1 around 2.5. This indicates accurately the self-activation of X1. Since we do know this vector field equation, then we can actually use it to calculate the Jacobian at any point in this data space, and then reveal the cell state dependent self-activation as a heat map. And we can also do the similar analysis for the Jacobian from like X1 to X2, and to reveal the repression from, sorry, okay, anyway, and then the repression from X1 to X2, as shown here. So now I told you some theory, like so I'm gonna actually tell you how we can learn the equations and use it to make a gain biological insights. So as I mentioned, we use the dynamic systems view to understand the cell state transition, but we generally don't know this underlying gene regulatory network. So we would love to actually learn it. So fundamentally, to learn this this network or this vector field function, this is a machine learning problem where we have some velocity data, which include the current cell state X and also some local dynamics or dx over dt. And then this kind of data can be generated with the conventional STARmap or the metabolic labeling enabled STARmap. The later ones will give you better velocity, as I mentioned before. So, but we will take just pairs of data and then learn using the machine learning algorithm to learn vector field function F, which then we can use it to predict the trajectory of each individual cell, and also making non-trivial predictions, as I will explain next. So our method again is very general, and here I'm going to just quickly show you how we can use this method to reveal two additional biological processes, like the neuronal activation and also the cell cycle progression. Mathematically, the vector field learning is trained to searching for optimal function in a vector field functional space that fits the input data space and using this loss function. And here, as you can see, among the G, H, and Q, these three functions, the H give us the best fitting. So then we will select this as our optimal vector field. And then the vector field mathematically is represented as a summation of a series of base function, which we implemented as a Gaussian kernel multiplied by some coefficient vectors. And of each base function is associated with a landmark like x tilde 1, x tilde 2, and so on. And then we can sum up all this base function to get this final continuous and analytical vector field. Because we do know the form of this vector field, we can actually use it to analytically calculate the RNA acceleration, Jacobian, curvature, core, divergence, and the others. And this analytical calculation of this differential geometry quantity is incredibly fast. We can, for example, calculate the RNA Jacobian between thousands of genes in thousands of cells in just a matter of minutes. So, as I mentioned, we can calculate this differential geometry quantities, and each of them actually have deep biological meanings. So, as I mentioned, the Jacobian can tell you the gene regulatory network. For the divergence, it can tell you the source and the sink of the cell space. And then for the acceleration, it can tell you the early driver. For the curvature, it can tell you the genes that are responsible for the cell fate amplification, and also for the core, it may potentially related to the cell cycle. So here I'm gonna show you a few examples of how we can leverage this quantity I just mentioned to gain mechanistic insights for different dynamical processes. So let's first talk about the divergence. So divergence, as I mentioned, it's actually defined as the trace of the Jacobian. So if you have negative value, it indicates the local flow has a convergence. So this may indicate a sink or potential terminal cell type. And if the divergence is positive, this may indicate the local flow has some like dispersion. And so this may indicate the salt or potential progenitor cell. And indeed, if we when we apply this method to a data set of zebrafish differentiation, you can see the window progenitor cells, the divergence is significantly much it's positive, and also the surrounded terminal cell type actually have negative divergence, correctly indicating divergence can be used to distinguish progenitor cell and also a terminal cell type. So next, we also gonna also show you. Yeah.
A quick question. On the previous slide about the divergence, would you then go on to interpret like a region of positive divergence as a region where there's high growth rate or like proliferation?
Proliferation. I think it may be able to do that, but I'm more thinking about the differentiation, like the cell is moving away from the progenitor or sedan cell state and going to different terminal cell state. That kind of like a force-driven cell moving away, the progenitor state can be identified by divergence. Another, yeah, it is in my experiment.
I think you're right. Like I think if there's high proliferation, but just one direction that they're differentiating.
Yeah, proliferation may be more related to the cell cycle because we have the cell cycle going on, and that may be related to like the core actually. Yeah, please, another question in the back. Yeah.
What dimension do you integrate the vector field in?
So the vector field, by definition, can be defined into any space.
Yeah.
It can be a two-dimensional space, like a universe space, or PCA space, like a top 30 PC, or you could even do in the original gene expression space. And the general way we do it is in the PCA space because it reduces the noise, but also you can go back to the original gene expression space. So generally, it's PCA space. But when you calculate these quantities, I didn't mention it, but for like acceleration curvature, Jacobian, they actually on the gene space, you actually able to know the acceleration, just like the expression for each gene. You can actually know the acceleration curvature for each individual gene in each individual cell.
So then to obtain these pictures, you project the vectors from the PCA space down to the...
So what I did here is I just calculated the known of the acceleration vector. And then this is sorry, no, this is divergence, but for the latest one, I'm gonna tell you later. We can actually calculate the acceleration for individual genes.
I see.
Yes.
Just a quick follow-up from that. Yeah, so this issue of dimensionality is quite interesting because I would guess that the dimensionality in different points of this state space is kind of different. Like there are different numbers of like gene modules that are active in the progenitor cell and axes of variation that are like.
Yeah, that's right. But I think you probably are more talking about the effective genes, right? So the dimension for the system is the same because you have the same number of genes, so the dimension will be the same. But if you are thinking about the effective the genes that are activating, yes, they're different. Some genes will be only expressed in this cell type and not in other cell type. But that this kind of phenomenon doesn't affect us because we can quantify that in our model.
I guess I'm just trying to understand because I mean if you imagine the the cells living on a submanifold of gene expression in space.
Yeah.
Right? The dimensionality of this submanifold is probably different at different
Yeah,
points.
but as I said, just some genes are not changing. Yeah. In vector field, the vector field is defined in the entire gene expression space. If you see this in these regions, if some genes are not changing, it just means the velocity for this gene is 0. And when you perform acceleration and curvature analysis, they are all 0. Yeah.
Okay. Thanks.
Okay, so I think for the sake of time, maybe let's move on. And potentially, I don't think I can go to my spatial study, but if you are interested, you can also ask me later. Then for now, let's move on. So another, as I mentioned, another weak core property is the acceleration. So here, I'm going to use this P1 GALA1 gene expression. It's a good example to show how we can use acceleration to reveal the earlier drivers. And here, what you're looking at is a path that predicts the vector field from the HSC to the neutral field cells, and SPA1 or GALA1 are two key regulators for the hematopoiesis. And then, as I just mentioned, although this vector field visualized in 2D, we are actually able to go back to the original gene expression space, so we can actually take the acceleration and velocity and also expression for each individual gene. And then you can see, interestingly, for the acceleration of SPA1, you see the peaks it has a peak at 21 hours, and then the velocity peaks at a much later time point, like 47 hours, and the expression is much later, like 70 hours. So this indicates that the acceleration can be used to predict the earlier driver that happen much earlier than the gene expression actually turning on. And then another very cool thing about Dynamo is that you can potentially get effective genetic genome-wide kinetic experiment for one single cell experiment. And here again, I'm going to use this SPA1 GALA1 as an example. But again, this kind of analysis can be applied to any other gene and any other systems. So it's really general. So let's first look at the string name plot for these two genes, SPA1 and GALA1, and you can see that there is a very nice specification between SPA1 and GALA1, and the high expression of SPA1 GALA1 will lead to monocyte neutrophil cells, where the high expression of GALA1 actually move towards to like metakaryocytes, erythroid, and basophil cells. And when we plot the Jacobian, so each dot is a cell, and the color is Jacobian from SPA1 to GALA1, and you can see that with the middle box is A, there is a strong strong negative value indicated the repression from SPA1 to GALA1, and this kind of repression gradually attuned when cell get into the mature cell state. So this again reveals this mutual inhibition between these two genes. And then we can plot the probability density plot of the Jacobian under a different P1 GALA1 expression. And then you can see the repression from SPA1 to GALA1 has this kind of shape. It's a function of SPA1. And where the repression from GALA1 to SPA1 is linear, and there is no threshold. And then we can actually use like the here equation to fit the kinetic parameters for this system. And then you can see, interestingly, the fitting gave us like a here coefficient n around 2.3 for the repression from SPA1 to GALA1, and but that value from GALA1 to SPA1 is actually 1. This indicates that the repression from SPA1 to GALA1, which has a ratio, may actually remove the damage where the repression from GALA1 to SPA1 requires potentially only monomer. And indeed, previous literature shows that the repression from GALA1 to SPA1 has no threshold. You can see as GALA1 increases expression of SPA1 actually linearly differs. knowing that this kind of experiments actually takes years to complete with just one pair of interaction. So we are really excited about the possibility of our method to use to predict the genome kinetic parameters with just a one single cell RNA-seq experiment or one kinetics run. So again, I wanted to emphasize that the real innovation of Dynamo lies in the fact that it marries the scalability of machine learning with the integrity of dynamic systems and also differential geometry. When with the machine learning approach, then we can use it to learn the vector field in high-dimensional space and also for many cells. And also with on the other hand, with the dynamic system approach, you can use it to identify the attractor, attractor-based subtle point separatories, and also going beyond the RNA velocity to RNA acceleration, curvature, divergence, and also other terms. And this kind of marriage between machine learning and the dynamic system is really cool because it reconcile the black box from the machine learning with the white box from dynamic system. So it gets a good benefit from both fields. And also with the vector field, let's dive into some fundamentally novel predictions that the pseudo-10 or RNA velocity cannot achieve to gain mechanistic and predictive insights of self-fit conditions. So here I'm going to use the human hematopoiesis as our system, but again, this same analysis can be applied to other systems. And here I'm going to quickly show you that we can use this approach to make non-pure predictions. Like for the maybe okay, because I only have 5. How many minutes? I have 5 minutes. 17 minutes. Let's slow down. Start at 50. The schedule 12 minutes to start at 50. Okay, great. I'm so important. All right, maybe then take it easy. All right. So, so in over the past decade or so, people who study human hematopoiesis find that the megakaryocytes always appear much earlier than other lineages. But we actually don't really know what's the underlying molecular mechanism driving this early appearance. And here I wanted to leverage Dynamo's predictive approach to tackle this problem. And then, so I started with learning the vector field of the hematopoiesis data set we generate, and then we identify with attractors of this system, each of them corresponding to the stable cell type. And then I use, and then I try to calculate the time from the HSC to each of the mature cell type. And interestingly, you can see we indeed reveal the megakaryocyte at a maturation time of 31 hours. Because we do know how much because we do know how much time we label the cell, we actually obtain absolute velocity, and this time is actually consistent with other single cell transplantation experiments. Next, in order to understand the underlying molecular mechanisms, and then look at the gene expression of FLI1, masked regulate of megakaryocytes, and also the gene expression of KRAS1, masked regulate of erythroid cells. And then you can see the FLI1 tend to have a high expression already in the progenitor cells, but the erythroid cell only have high expression in the erythroid cells. And
so this middle panel that you're showing is like titled like absolute velocity. Is it the geodesic time that it takes to go from the stem cell state to any of these different?
Yeah. So the method that we are using is called the least action path. Basically, you give like some kind of initial state and some terminal cell state, and we actually explain to you, and then we optimize the path that has the smallest action. it's similar. I mean, it's related to your distance, but it has some like optimization behind it.
Okay, thanks.
All right.
I was confused because it's absolute velocity, but it's mentioned times. So
Yeah, absolute velocity. Just saying that we use labeling data. So
right.
you have actual time instead of relative. Like if you use SC value or velocity, this you cannot get the actual time. You only get the pseudo time. Yeah.
Thank you.
So yeah, then we perform this Jacobian analysis and reveal there is actually repression from free one to KLF1 across all the cells. And then additional Jacobian also shows that free one actually has a self-activation. So what this means is that the free one tend to have like a self-activation. So it will maintain high expression of free one in the progenitor cell. And this high expression is also maintained through the repression to KLF1. So the HSC then will bias toward to become megacases, and the megacases will then appear much earlier. And now I'm gonna show you some non-trivial prediction Dynamo can do. So ever since this discovery of the cellular, the reprogramming, there has been kind of a fundamental question in biology to like predict what's the optimal TF combinations to convert one cell type to another. So previously, I used that's actually a paper I published 10 years ago. I used this least action path, which is a fundamental concept in species, to predict in simulation the optimal developmental differentiation and also transdifferentiation path. Now, because we actually have a equation learned directly from data, so I wanted to one of the ways that we can actually also use the equation, such an equation to perform the lab analysis. So in the context of human hematopoiesis, we do know a lot of like differentiation and also reprogramming and even transdifferentiation events, and we also know the genes associated with each of these conditions. So the idea here is that we wanted to first validate our prediction on these cases, and then making normal predictions. Intuitively, the lab method is just saying, let's say if we wanted to convert from the HSC state to the megakaryocyte state, in principle, there are infinite number of possible paths. You can either go to this path, or the other path. But some paths will be more likely than others. So here we can see energy. Under this optimization defined by this action function, we actually find that this first path has the smallest action, and so it becomes the least action path. So and now I'm gonna actually show you how this method applies on the real data, and on this plot it reveals the lab from the HSC to each of the terminal cell types. Interestingly, you can see this is actually not the shortest path on this vector field. It actually follows the curvature defined by the vector field. This is because if we follow this string line, this action turn will be 0, so it will have the smallest action. And again, because although we visualize this in 2D, but our operation can be go back to the original gene expression space, so we can use it to reveal the gene expression changes along this path for different genes. So here, each column is a different gene, and we are visualizing the least action path, the gene expression change along the least action path from HSC to I think the base of the image. And the important thing is that we can also start to identify genes that make the biggest contribution along this path. So here we use a priority method to calculate the priority score and identify correctly the known key transcriptomic factors like GALA2, 1X1 as the key genes for this transition. So here I'm gonna quickly show you the priority score of all the known transcriptional factors of each of these reported reprogramming and transcription events has mostly like a score above 0.8, with 1 is a perfect score, and also a rock curve analysis of all these known conditions has a AUC of 0.85, indicating the high predictivity of this approach. So given the success on the known transitions, it will be, so here I'm gonna actually show you the prediction of the top 3 genes of the entire transition matrix. And also their kinetics amount is predicted this action path. It will be a very interesting future direction to validate these genes and also the kinetics from the prediction, especially for these yellow boxes, these are unreported conditions. So to further showcase the predictive power of our approach, we then designed the in silico perturbation method. Here I'm going to highlight one perturbation prediction on this PU1 GALA1 network motif. So because PU1 GALA1 the self-activate and mutually inhibited, so it's like a seesaw model where if you separate the expression of GALA1, then this balance will broken and cell then will move towards the GMP lineage. Indeed, if we run this, if we apply this equation and run the simulation with Dynamo, then you can see for the cells at initially the evolutionary MAUP lineage, after you perturb you remove the GALA1, the cell indeed move away and move towards to become neutrophil monocyte cells. Also, our method is very highly efficient and combinatorial and genome-wide. So potentially you don't actually need to do any experiments that make the genetic perturbation. This will provide an important complementary method to the genome-wide perturb-seq that Ruben gonna talk next. So from the above slide, I hope I convince you that after 10 years, this is the paper published 10 years ago. My dream of like predictive modeling of genomic dataset is really final. It's really realized in this Dynamo paper, and I also addressed the questions in the beginning of my talk, like, and show labeling data give us the best data for learning the equation, and then we can use the kernel method to learn the equation, and then we can use this equation to make predict the future cell state and gene regulatory network, and even making like this action path and the in silico perturbation prediction. And how many minutes? Okay, and so I'm gonna probably quickly go through my because I don't really have time. Yeah, so I'm gonna actually skip all the published work. I just tell you that this Stereo-seq is awesome because it can increase the spatial resolution field of view, and because also it has very high resolution, we can achieve single cell segmentation. Then we can do the segmentation for entire embryo, and then visualize this kind of to visualize the cell type distribution of the entire embryo. And importantly, we can also, because it has a high spatial RNA capture, this method can enable us to perform RNA velocity in situ. And here I visualize two neurogenesis process. One is tangential migration, and the other is radial migration. And next,
Laboratory label.
No, this is just use simulation answer. Okay. Of course, that's a future direction, and we are thinking about the campaign and labeling and spatial. Yeah. So I think I only have maybe 1 minute. I'm gonna quickly tell you that now with this technology, how we can actually achieve single cell resolution and 3D, because most of the spatial transcriptomics technology currently only just 2D. But how we can move away from 2D to 3D spatial data for a whole tissue and organ. So to address these challenges, then we recently developed a new tool. I'm not going to talk the details, but with this method, we are able to achieve 3D spatial transformatomics. Here I'm gonna just quickly show you that leveraging the Stereo-seq, the whole field of view, and you can perform serial section of the same embryo. Here I use the Postdoctoral embryo as an example. And then after you do the slicing, you can do the Stereo-seq, and then you can computationally enable these slices to reconstruct like a 3D point cloud, where each point corresponds to cell, and the color is cell type. And then you can take this data to do a 3D mesh to create an in silico model of the embryo. And then you can also try to look at the 3D gene expression. We first create like a 3D work cell, and then we use a deep learning method to map the gene spatial coordinate to the gene expression. And then once I have this model, then you're actually able to perform any arbitrary selection in the selection on the AP axis or the dental voice, the ventricle dorsal axis, and to know the gene expression gradient on the 3D space. And we can even use this to like create the tissue level mesh and understand the interactions between different organs in 3D and perform novel morphometric analysis. So with that, I'm gonna thank Jonathan for giving all the support during the past few years, which really made this work possible. I also want to especially thank Jianfa and Yan Zhang, who made an incredible contribution to this work. Without their help, this is not going to be possible. For the sake of time, I'm gonna probably stop here and just tell you that now we are actually trying to extend Dynamo and also spatial to like a software ecosystem called Explorer. So for the purpose of building the foundations of predictive and mechanistic single cell and spatial genomics research. So if you are interested, feel free to connect me, and I would love to talk more.
Xiaojie for the fantastic talk.
Thanks again. Yeah.